Good morning, guys. Can you hear me?
Yes.
Yes.
All right. All right. Hope you had a good weekend. What's Monday, September 28th? We are more than a month into the fall semester. And hopefully we've learned a few things about biochemistry, right? So we finished in chapter 5 today, protein transport. And then we're gonna have our first test on Wednesday. So I already made the test. Test is already sitting on your Canvas site, right? So you're just gonna have to log in on Monday morning at 9 o'clock, right? It's gonna be a 55-minute exam. So most of the questions are kind of yes or no. So it's not a difficult exam in terms of time, right? So it's not gonna take you much time. So you should get done pretty quickly. As I mentioned to you, not too much math. There is a little bit of math, but so you may want to have bring with you or have nearby a calculator, right? It doesn't need to be a scientific calculator. There are no logs or anything. So just a basic arithmetic calculator. And so essentially practice test is very similar to the practice test, and essentially pages from the textbook just for you to review, right? So presumably you don't need to read all this for the first time in a few days before the test. So hopefully you've been reading this all along. So this is just a review, right? These pages. And do you have any questions?
I have a quick question.
Yeah.
For the exam, oh my gosh, I lost it. I'm sorry. But for the exam, will we need to log in after if we finish the exam to Zoom? And should we look at the questions at the end of the chapters for a possible study guide for your exam? Or you think those would be too complicated because they do include a lot more math than your questions would?
Yeah. So any kind of practice is good. So just keep in mind that if the questions are more complicated, it's not necessarily a waste of time for you, right? Because you're learning a lot by doing complicated questions than by simple questions. So even by doing math, you're learning things, right? So you may have to use a calculator more, but you reach conclusions. You learn something from these problems, right? So by doing problems, you learn from them. That's why I've been pretty liberal with the sapling, because sapling, by doing sapling problems, you learn tremendous amount. It's not just for you. It's not just for me a way to test you, right? So you work on problems, then you read through solutions, and you learn tremendous amount. So don't. If you have complicated problems, if the choice for you is to do them or not to do them, if you have time, I would do them. So even though the exam it's better to have to be prepared and have the exam to be easier than you thought, right? Than the other way around. So.
Okay, thank you.
Anything else?
How many questions is the exam?
Exam is 30 questions, 55 minutes. So you have about 2 minutes per
Perfect.
question.
Perfect.
Some of them will take you probably 5 seconds. Some of them will take you a little longer. So I tried to, to prepare this exam. It took me quite a bit of time, specifically because I'm not using in this class any kind of proctoring service, right? Which in most cases works quite poorly from what I hear from other professors. So I try to come up with questions that you cannot Google easily, right? So questions that you have to actually think and generate your own answer. And without knowing the material, it would be very difficult to generate an answer. So so if you are very honest and you're not gonna Google things, and so I think you should be quite quite happy with the fact that actually your classmates will not have the opportunity to do that. So okay.
Are we real quick, Doc? Are we required to log into Zoom while taking the test?
No, just Canvas.
Okay, thank you.
Just Canvas. You can. Well, I'll be around. Well, you can email me quickly. I'll be on my email, so I can respond to your emails very quickly.
I can't remember if you said this before, but how many questions are on the test? You said it's 55 minutes, but
30?
3 questions?
30?
30. Oh, okay. Thank you.
Yeah. Mm-hmm. Anything else? All right. Well, email me if you have a general question. I would prefer if you place it in a discussion. If you have a more personal question, then you can email me. All right. So let's go to the material, and let's finish chapter 5. So we are being recorded. I didn't forget that. Share screen. Okay. All right, so we talked about how oxygen binds to the iron, which is part of the heme, right? We talked about the conformational change and the T state and the R state, right? Remember tense and the relaxed state. So in the tense state, remember that oxygen affinity for the hemoglobin is low, and that's what is preferred in the tissue where the oxygen needs to be released. And the R state is the state where in which the oxygen affinity for hemoglobin is high. And so that's a state where that hemoglobin adopts while being in the lungs when it needs to bind more oxygen to bring it inside the tissues. So so we talked about these two states. So that's one way to regulate. Another one is oxygen affinity for hemoglobin is also regulated by acidity. So there's a pH effect. So you can see here in this graph, right? So remember, this is partial pressure of oxygen, right? Partial pressure of oxygen versus site occupation on hemoglobin, right? So 0.5 means half of the sites on hemoglobin are occupied by the oxygen. So tell me, let's answer this question. Does acidity increase or decrease the KD? Well, first of all, KD. Remember KD? That one very important unit, one very important measure of affinity of ligands for the receptors. Remember, it's called dissociation constant. So you had it on sapling all over the place, and it will surely be on the test. So dissociation constant. So when your binding is tight, is the dissociation constant high or low?
Low.
Low. So so you'll be looking for so the tighter binding should give you low dissociation constant. So tell me, is the does the acidity result in low or high dissociation constant?
High.
Correct. So remember, one definition of this dissociation constant is the substrate concentration, or the partial pressure, in this case, of oxygen, at which half of the sites are occupied, right? So you can see here. So this will be, this results in this KD. Now at pH 7.4, half of the sites will be occupied here. At pH 7.6, half of the sites will be occupied here, right? So you can see that as you decrease acidity, as you raise the pH value, the KD keeps dropping, right? So what this means is that high acidity, which is for the most part present in the tissue, right? So in the tissue, acidity is high. In the lungs, the acidity is low. So in the tissue, where acidity is high, the KD is high, which means the oxygen doesn't bind to hemoglobin strongly and is released by hemoglobin into the tissue. So that's another way hemoglobin has evolved. And so depending on the tissue, on the acidity, it can bind oxygen more tightly or less tightly, and in the tissue, it binds it less tightly, and the oxygen is released. So the question is, why is the acidity higher in it? So the pH is lower and acidity is higher in the tissue. So remember, the tissues are going to be actively metabolizing, and they generate lots of H+, because they have obviously they burn that food, right? They burn the nutrients using the oxygen that hemoglobin brought to begin with, and generate carbon dioxide. Now, what happens to carbon dioxide in the tissue? Most of it, now there's an enzyme, carbonic anhydrase, very critical enzyme in the tissue and in erythrocytes, primarily in erythrocytes, actually. So basically what it does, it makes carbon dioxide react with water to produce bicarbonate, tissue bicarbonate, and protons, these two. Okay. So the question is, why does it do that? So now imagine if you have lots of carbon dioxide produced in the tissue, what will happen? What will happen to you? Can you imagine all the gas produced in the blood vessels in the tissue? It will be bubbling all over the place, right? So what it does, basically, it reduces the amount of gas in the blood vessels in the tissues by reacting that with water to produce a salt, which is going to be soluble in the bloodstream. And as a result, it will produce protons. And as we know now, the protons we just saw in the previous graph, hemoglobin affinity for oxygen depends on the pH. So H+ will bind to hemoglobin, and it will stabilize it in the T state, dense state, low oxygen affinity state. And here, the way it's going to work, it's going to protonate histidine 146 of the beta chain, which then forms a salt bridge with aspartate 94. And this salt bridge will lead to the release of O2 in the tissues. So this is known as Bohr effect. So please remember that. So the scientist who discovered this, his name was Bohr. So the way this actually will work is, now let's look at this. So let's say you have histidine 146. So this is histidine. One 46. Of the beta subunit. Now, in the tissue where the acidity is high, H+, in tissue. There's a lone pair on this nitrogen. Remember we talked about in histidine, when you have the imidazole ring, I asked you already before in this class, which nitrogen will be protonated? And you told me it was this one. Anybody remembers why?
It's more conjugated.
Conjugated, there's,
It has a lone pair too.
well, this one has a lone pair too. So why is this going to be protonated, not that one?
The
Would it-
lone pair on this nitrogen on the left doesn't contribute to aromaticity.
That's right. So this lone pair is part of the aromatic system. That's right. And this lone pair is not. It's actually perpendicular, right? So this lone pair lies in the plane of the screen, and this lone pair is actually perpendicular to the plane of the screen and parallel to all the p orbitals of the pi system of the imidazole. Right, so this lone pair will reach over, grab this proton. So we'll have our histidine 146. Remember that means it's amino acid 146 from the amino terminus of the beta chain, right? So it's a beta chain. Beta chain. So we have this nitrogen. It's going to be protonated. And it's going to have a positive charge, right? So now this, once it's protonated, it can form a salt bridge. It can form a salt bridge with aspartate 94. So also from the beta subunit. So here we have an oxygen. Negative charge. We'll just put R here and say that this is aspartate. (94). And as this happens, the result is the transition. Now, once the salt bridge forms, the result is the transition. Yeah, we have some other species listening to the lecture. The result is the
Sorry.
transition. No problem. The result is the transition from the R to the T state. R to the T state, and as a result, O2 release. O2 release. So remember, this is Bohr's effect.
Just to clarify, the formation of the salt bridge is what is like initiates the transition from R to T state.
Yeah. So so obviously there's a conformational change within the protein, right? So when this nitrogen has no proton, it has no business interacting with aspartate 94, right? You have a negative charge here, you have a lone pair, electrons hate each other. And so clearly this nitrogen and the oxygen of the aspartate have a significant distance between them. And so the protein is in a totally different conformation in the R state. Once you protonate this nitrogen, then now they start interacting. They come into close proximity. The protein changes its shape from R to T, and that results in the oxygen release. This makes sense? I think it's very neat how hemoglobin evolved to have this, not only to be an allosteric protein in terms of cooperative oxygen binding, but also in terms of acidity. But that's not the end of the story. It turns out that carbon dioxide, so a lot of it is going to be hydrolyzed by this enzyme carbonic anhydrase, right? Carbonic anhydrase given by carbonate and protons. But there's another way to remove carbon dioxide from the tissue. After all, and the tissues have to get rid of carbon dioxide, right? So it's all very well that the hemoglobin brings oxygen, but how do these byproducts of the metabolism, how those are removed? So so that's one way. So another way is direct binding of carbon dioxide to hemoglobin. And it turns out that about 20% of this carbon dioxide will be exported in the form of a carbamate. Carbamate on the amino terminal residues of each of the polypeptide subunits. So that's how it was written in the text. The way I understand it, so each of the polypeptide subunits probably means that, so we have 4 polypeptide subunits, right? And so, so there are 4 sites for carbon dioxide on hemoglobin to bind to that, right? So these are primary amino groups. And you can see the way it's going to work is that you have again, lone pair in nitrogen. You see it's all organic chemistry. Attacks the carbon dioxide. It's like attacking a carbonyl group. The electrons move over here. Electrons will move over here. So let me draw an intermediate so that we know exactly what it looks like, so that we can utilize our organic chemistry knowledge to understand this in more detail. And so the nitrogen will be positively charged, right? Nitrogen is positively charged. And I'm just gonna have a wavy line here showing that there is more stuff here. We're just not gonna draw it. And then what happens? And then a base, right? Which will probably be water. Which will probably be water. Will come in and grab this proton. Electrons move over here, and that gives rise to the carbamino terminal residue. Okay. So and then so hemoglobin will take this to the lungs, right? And what will happen in the lungs? In the lungs, the reverse. will happen, right? So once the carbamino adduct, once this is brought to the lens, a reverse process will happen in lens. Reverse process. Okay. One important thing here is also, look, when the carbon dioxide binds to the amino terminal residue, there is also a proton produced, right? So the water will grab this proton, and you can see here it's released in the form of hydronium ion, of course. But the formation of each carbamate will each a proton that can contribute to the Bohr's effect, right? So this will even increase the acidity in the tissue even higher. So carbon dioxide, so it's another way for carbon dioxide to increase acidity in the tissue is through by reacting with the amino terminus of the hemoglobin. And this carbamate will form additional salt bridges, stabilizing the T state. So let's do that. So let's say we have what amino acid. Let's say we have lysine. Lysine, some kind of bridge. So side chain lysine. Where the amino group is protonated. Right? And so this will form a salt bridge. With the carbamino hemoglobin and terminus. And again, the result of that is this transition from the R to the T state. Okay, so both high acidity and high carbon dioxide concentration in the tissue will reduce the affinity of hemoglobin for oxygen and result in its release in the tissue. Any questions about this? I think it's very cool how this all works. All these processes allow for hemoglobin to bring oxygen from the lungs, to have high affinity for oxygen in the lungs, and reduces affinity in the tissue.
I have a question. Does this have anything to do with nitrogen staying in the tissues, like when you dive for too long?
Nitrogen
Is that a part of this process?
Nitrogen staying in the tissue, what?
Does this have any part in the process when you get the bends, when you're diving for too long? Nitrogen stays in the tissues?
You said when you die?
Like scuba diving?
Scuba diving.
Yeah.
Oh, when you dive.
I know, I can put it in the discussion.
Yeah,
I was just wondering.
Yeah, I don't know much about that. So
Okay.
why don't you put in a discussion and we're all gonna look into that.
Got it.
And that's what discussion is for. All right, but that's not the end of the story. There is more. It turns out there is another molecule, 2,3-biphosphoglycerate. It's shown here. So glycerate is the carboxylic acid, which has 2 hydroxy groups. So this is carbonyl, right? So this will be glycerate. Glycerate. And then if you make a phosphate ester with the 2 oxygens of the glycerate, then it becomes biphosphoglycerate. So it's actually quite an abundant molecule. So especially present in erythrocytes in millimolar concentrations and produced from intermediate in glycolysis. Small negatively charged molecule binds to the positively charged central cavity of HB, of hemoglobin. And it also stabilizes the T states. Well, in the compared with the oxygen on hemoglobin, we have 4 oxygen binding sites. For BPG, bisphosphoglycerate, there's only 1 binding site. 1 binding site, and it will basically move the hemoglobin from the R to the T state and reduce the affinity of all 4 of the oxygen binding sites. So there's a T state, and you can see here in the T state, there's a very nice cavity here. And it's usually when you see blue, that means is that we have amino acids such as lysine and arginine, which are positively charged. So why would you want to have a positively charged amino acids in the cavity where BPG binds?
Because it's negative.
That's right, BPG is negative. So BPG will be attracted to this binding site because there are nice amino acids which are positively charged. And so that's the T state. So T state has this pocket for it. Now, when it moves to the R state, this pocket disappears and BPG cannot get in. So in the R state, which has high affinity for oxygen, BPG cannot get in. All right, so here, let's see how BPG, why BPG is so important. So this is a very important graph that requires some work, right, to fully understand what's happening here. So it turns out that BPG plays a number of important roles. And one of them, for example, in adaptation to high altitudes, right? So you remember, for example, climbers who climb tall mountains such as Everest, right? They go to the base camp and they live at the base camp for a while. Can't you remember how long? A week, 10 days, right? Something like that. Anybody climbed Everest? No? To acclimatize, right? And specifically, now the people in general, some basic explanation for that. They're just saying that to acclimatize, to be able to climb such tall mountains, you need to have more erythrocytes in the blood, in principle, right? So the blood can actually extract more oxygen from the atmosphere, right? Even because the oxygen concentration is low at high altitudes. And specifically, you can see here, so this is partial pressure of oxygen in the lungs at the sea level. It's about, what, 13 kilopascals. Now, partial pressure of oxygen in the lungs at 4,500 meters is about 7 kilopascals. So about half of that, right? About half that. But this is what's happening. So the number of erythrocytes is important, but there is an even more powerful method for us to adjust to living or to staying at high altitudes. So if there is no BPG, in the blood, in the erythrocytes, then the all hemoglobin is going to be in the R state. And you can see here, it will bind oxygen very strongly at very low partial pressures of oxygen. All the binding sites will be immediately occupied, right? Even at the 2 or 3 kilopascals, you can see all the sites. So the theta or Y, we still haven't figured out why. Sometimes it's theta, sometimes it's Y. I need to look into that. So Y it's 1.0, right? And you can see here, so this is this would be if there was no BPG. If there was no BPG, you can see here we grab all this oxygen, put it in the lungs, take it to the tissue with the erythrocytes, but the erythrocytes will not release any of the oxygen in the tissue. So it's a waste, no use, right? And so that's the role of BPG. So at sea level, the concentration of BPG, which puts the hemoglobin in the T state, is 5 millimolar, and is this blue curve, right? So it's sigmoidal curve, as we talked about. So the sigmoidal curve, what this allows us to do. So in the lungs at sea level, right? So this is how it will basically almost all hemoglobin binding sites will be occupied with oxygen, close to 1, right? Now, as the erythrocytes move towards the tissue, and on average, in the blood vessels, capillaries in the tissue, the partial pressure of oxygen is about 4 kilopascals. And you can see at this oxygen pressure, about 60% of all the binding sites will be occupied, resulting in a 40% difference. And more exactly, it's 38% difference. So what that means is that when BPG is 5 millimolar, right, at sea level, 38% of all the oxygen from erythrocytes, from hemoglobin, will be delivered to the tissue. Not bad, right? Given the fact that basically we're dealing with the same molecule, hemoglobin, bound to oxygen in the lungs versus in the tissues, and 38% being released in the tissues is quite an achievement. But what happens as you move up in the altitude? Let's say you are at 4,500 meters. You start at this point, right? This is the partial pressure of oxygen in your lungs at 4,500 meters. It's going to be around 7. So you start here, and then you move to the tissues. Again, you go from 7 to 4 kilopascals. And here you can see the difference is only 30%, right? So you lose the efficiency. So there's 8% is lost. That's why you basically suffer from hypoxia, right? So normally 38% of oxygen is released in the tissue, but at 4,500 meters, if you're immediately exposed to that, only 30% will be released. And so what happens is that there is a way for this problem to be solved in biological systems, and specifically by the increase in the amount of BPG to 8 millimeters. So BPG, as you increase to 8 millimeters, remember it will even lower the affinity of oxygen to hemoglobin. It will raise the KD, dissociation constant. And so now we're dealing with this red curve, right? Red curve is to the right of the blue curve. And what it tells us is that the affinity for oxygen is decreased further. So again, we start, so here, now with the 8 millimolar of BPG, we start at this base, at this high altitude, 4,500 meters, right? But in the tissues, if you follow the red line, at 4 kilopascals, much less of the binding sites will be occupied because of the high BPG, right? And so you can see here the difference between this point, that point, and that point is 37%. So from 30%, we go almost all the way back to 38, is 37%, right? So by increasing the amount of BPG, we restore the delivery of oxygen from the lungs to the tissue. Any questions about this graph? I know you have to really look at it several times to really appreciate it. But it's a beautiful regulatory system, how by just a small increase in BPG, you can restore the oxygen delivery from the lungs to the tissue at high altitudes. So that's what the acclimatizing process is all about. That's why when climbers or people who live at such high altitudes, they have much more BPG in their erythrocytes than people who live at sea level.
That's from the lungs to the tissues.
From the lungs to the tissues, yes. Mm-hmm.
So to clarify, the BPG will help take more oxygen to the tissues whenever you're climbing, for example?
Right, so more oxygen will be, the difference between the oxygen saturation of hemoglobin in the lungs and the tissues will be restored to the normal one, which is 38% or 37%. So yeah, so more oxygen will be delivered to the tissue. Mm-hmm.
Okay, thank you.
All right, there is one other aspect of hemoglobin and oxygen delivery we need to briefly talk about. There is a genetic disease known as sickle cell anemia. Sickle cell anemia, which is a mutation. It's a mutation in the beta chain of hemoglobin, specifically glutamate 6 is mutated to valine. Okay, so the mutation has to happen in both alleles. Remember, we have 2 copies of the same gene, right? One from the mother, one from the father, and they're called alleles. Both alleles. So if you have this mutation in both alleles, then your hemoglobin is defective. Your hemoglobin is defective, and specifically, let's look at this picture. Basically, it changes the shape of erythrocytes. Let me let me try to describe where's the. So I guess we don't have the picture of the normal erythrocytes. So normal erythrocytes are just like discs, like this, right? That's their normal shape. In sickle cell anemia, when you have this mutation of glutamate 6 to valine, you have these variably shaped erythrocytes, right? You can see they adopt various different shapes. And which one is sickle? This one is sickle shaped?
It's the one above and the one on the bottom.
This one and this
Yeah.
one.
Yeah.
Yeah.
So because of the shape, that's how the disease is called, sickle shape anemia. And so untreated, so if when both alleles are affected, the individual is known as a homozygous, which basically means that the mutation occurs in both alleles, right? So it's homozygous, both genes are the same. And so those individuals, if they're untreated, they generally die in childhood. But the interesting thing is that heterozygous individuals exhibit a resistance to malaria. So in fact, only in heterozygous individuals, only 1% experience the symptoms of sickle cell anemia. Same. Of this disease. But why this disease is prevalent in many African countries where we have malarial mosquitoes, right, that carry the parasite, is because the individuals, it gives them almost full immunity, or not full immunity, but significant resistance to malaria, right? And because only 1% will experience the symptoms of the disease of the sickle cell anemia, so it's a sort of balance between the two, right? So it's all about the evolution, the fight for survival, right? So so this will diminish. This will increase some people suffering from sickle cell anemia, but it will decrease people who will contract malaria from the malarial parasite. So the way this works is this new valence side chain can bind to a different hemoglobin molecule to form a strand similar to the amyloidogenic proteins. So just think about it. Glutamate is a charged amino acid, right? Valine is hydrophobic, so it's sticky. So the key word here, if you remember, if you want to remember the cause of this disease, remember the word sticky. Sticky, that means that valine, right, is hydrophobic, much more sticky than glutamate, which is negatively charged. And so what happens is that, so this is, let's say, this is normal hemoglobin A, right? So there are 4 subunits. Now, this is hemoglobin S. And in hemoglobin S, I guess this, what is shown here in this figure, this beta 1 portion here is kind of round, right? Whereas this one is kind of flat. And so this is where this valine is. And this is where this glutamate is, right? And so the valine will create a surface which will become sticky. And so different molecules of hemoglobin will stick to each other, and they will create interaction between the molecules, form some kind of strands, fibers. So this will crystallize. So the hemoglobin normally is a globular protein, right? It's water soluble. But once it's adopts these fibers, it will crystallize, it will fall out of the solution and form sickle-shaped erythrocytes. So yeah, that's one genetic disease coming from a mutation in hemoglobin. Any questions? And One last thing I want to say, which we didn't have really a page for it, but maybe I'll just do it here. There is also another difference in hemoglobin between fetuses and grown and individuals. So fetal hemoglobin has 4 residues, 4 subunits, but instead of 2 beta, it has 2 gamma subunits, versus alpha 2, beta 2 in adults. And it turns out that when you have gamma subunits, this variant of hemoglobin, it has high affinity higher affinity for oxygen. So what does that do? What do you think?
It allows better oxygen transfer in the placenta.
That's right. So better oxygenation of the fetal blood, right? So the oxygen can be extracted from the mother's blood, and more of it will be carried into the fetal blood. So sounds good. So, but this one is, you see, this one is beneficial mutation, right? This one is adverse. This one causes disease.
How do they like lose their gamma when they grow up, or how does that work?
Yeah, so I guess there are genes which are present. So basically, it's there must be genes for the gamma subunits, must be genes for the beta subunit, and those must stay in us for the remainder of our lives, right? It's just that different genes are expressed at different periods in development, right? So the gamma subunit expression must be suppressed as the fetus develops into a or comes out of the womb, right, and starts to breathe on its own.
Okay.
So it's a gene expression regulation, all about that. But the genes must be present throughout the person's life. Okay, if there are no more questions, now there are a few more things to read, which is just very descriptive to read. So these are pages. So if you read about the immune system, read about the immune system, pages 174 through 179. So that's the immune system. Basically, it's covered in these slides. There's not so much material here, just for you to familiarize yourself what the antibody looks like. And the reason why it's discussed is because antibodies are globular proteins. And the second topic to read again, that would be pages 179 183. So again, this is very descriptive. There's not really much to understand there. It's just how the antibodies work and how the muscular system works, right?
So are we responsible for finishing this chapter for the exam?
Yeah. Mm-hmm. Yeah, you have some problems in sapling on those. But I think if you understand the material on these slides that we have, that's all you really need. Okay. All right. Yeah.
I had one question. So what happened if we encountered like a technical issue in Canvas? Like let's say one of our internet drops.
Well, you have to take all the precautions, right? So make sure it doesn't. So find a most reliable station for you. Now, if you're not sure about your home internet, you can always go to campus and take the exam there, right? So that is rarely interrupted.
Okay.
So well, if worst comes to worst, we'll figure something out. But please make sure you do everything possible that doesn't happen.
Yeah. I was just worried about it wasn't the internet problem because on my previous exam for Canvas, it was the website's problem because they were having an external error. That's what I was worried about.
Well, Canvas is quite reliable, so hopefully it'll stay reliable on Wednesday. Yeah.
Okay.
Just don't use Safari if you have it. Like use Chrome or Firefox.
Well, I can't use Safari since I don't have a really MacBook, but yeah.
And you should be fine.
Yeah, Firefox is good. All right, guys. I'll be accessible by email and discussion. Talk to you later this week.
Thank you.
Bye.
Thank you.